Okay, good morning or good evening, everybody, depending on where you are in the world. Thank you so much for joining this webinar. As you know, this webinar, the goal of this webinar is to cover what's new in release 42 in the various EuPathDB resources. So we will go through various different resources and highlight examples of different types of data and functionality. Before we go on and get into it, I'm joined today by Suzanne Warrenfeldt, who will also be helping me teach, and my name is Omar Harb, and we're both part of the outreach team for EuPathDB. If you have any questions during the webinar, feel free to use the raise hand option in your GoToWebinar panel, or you can just go ahead and type the question in the question section of your GoToWebinar panel. And we will monitor those questions and try and answer them either verbally by voice, or we will go ahead and type an answer depending on what kind of answer is required. And all of the questions are visible by everybody, and the answers could be visible to everybody as well. So depending on how useful they are, we can make them visible to everybody or just answer privately. And if there's a question that we need help understanding, we might ask you to go, or we might unmute you so you can actually provide the question verbally. Okay, so as I mentioned, this webinar is, the goal of this webinar is to cover what's new in release 42. I'm looking right now at the EuPathDB homepage, and as you know, EuPathDB includes multiple different genomic and functional genomic resources. We have a news section on the left-hand panel here. This is available on all of our sites, but if you go to the parent EuPathDB site, you can quickly go in and choose the organism or database that you work on to see what's new in that particular resource. The first step that we're going to do is we're going to go ahead and cover. So let me go to the news section in EuPathDB. So I clicked on this all EuPathDB news. And so this will open up a page that shows me all of the news, and you can scroll down if you want to the section that interests you. But importantly, what we will first cover is some of the new tools and functionality that are available in EuPathDB sites. So all of these tools typically, unless there's something very specific to a particular organism group, all of them are typically available for all of the resources. So it doesn't matter what we demo this on, you should be able to do the same thing in your favorite website. So I'm going to jump to FungiDB to highlight, the first tool I will be highlighting is the Genome browser. And after I'm done highlighting the Genome browser, I will turn this over to Suzanne, and she will highlight some of the gene page tools and functionalities. So I'm looking now, I have a tab open for fungidb.org. Again, as I mentioned, it doesn't matter which website you're on. I'm just going to go to a gene page. And so I clicked on this search for an ID. So this is an ID that I had that I used before, but you can put in any ID for an organism that you're interested in. This allows you to quickly go to the gene page. And here I'm at this MFS transporter gene page. And you'll notice here that there's a beta sign that shows you that JBrowse Genome browser. So for all of our sites, we have a browser that allows you to view data in a browsing manner. So you can look at the gene models, you can zoom in and zoom out, and you can show various other data. For example, you can layer on top of this RNA sequence data or other types of data sets. And currently we have something called a Gmod genome browser, and this is functions as such, but there's a newer version that's been around for a bit called JBrowse Genome browser, also made available by the same group, but it is faster and zippier, and I think will be much easier to use. So currently we're beta testing it, and we encourage people to start using it, and I will just highlight how you can access this. So you click on the link JBrowse Genome browser. This will load the Genome browser for you here in the view. It's fairly quick, in particular compared to the current Genome browser that we use. You can click and drag, and so this allows you to move up and down the genome of interest. You can zoom in, or you can zoom out, and so that works very nice. It updates fairly quickly, and then you can load and layer additional tracks. And so there's a little select track tab up here at the top left side. If I click on this, this will open up all the tracks that are available. In this particular case, I'm looking at a, if I go back to the browser, I'm looking at genes in Neurospora crassa. And so when I go and select tracks, all of these tracks are any data that's available for Neurospora crassa. And so I can go in now and quickly say, I'm interested in, for example, here are a bunch of data sets. Let's say I'm interested in, let's see, transcriptome induction by 5 crop straws. I click on this. It shows me all the tracks that are available for loading. You'll notice some are unique aligned reads. In this case, I'm looking at RNA sequence, and others are non-unique. So I can quickly even use this text box here and say, well, I'm only interested in unique ones. And so this will filter right here on the left, the RNA sequence alignment. I can click on unique, and now I'm only looking at the uniquely aligned reads. And so this is just a nice way to quickly filter your results. I can select all of them. So I selected all of them. You'll notice in the background here, these tracks are already loaded. So I can go back to the browser, and here are all the tracks available. And you'll notice, again, I can quickly go in here and say, well, I want to zoom in and see what's happening in this area more closely. So I just click and drag, and then this gives you, zooms into the region of choice, right? And so depending on the level of expression, you'll see that this gene obviously has a lot more reads aligned compared to this gene right here. Another interesting track that you may be, that could be of use to you is, let me go ahead and, or I just want to demo it here. This is already available. So I'm going to clear all the filters here, and I'm going to type intron. And so you'll see here predicted intron junctions. So what we do is we use all of the RNA sequence data that's available to us, and we predict introns based on reads that span an intron from exon to exon, basically. And so I can go in and select, for example, this one here, which is, let's take this refined one, but there's some nuances between these. So obviously, if you're interested in these, you can delve into them a bit more. But I'm going to pick this RNA sequence evidence for introns. This is coming from all data sets combined. And so I selected this option, and now I'm going to go back to the browser. It typically loads at the bottom of the page, so I can click and drag it up, because maybe I want to look at this close to the gene models, right? And you'll see here that there are some introns that are predicted based on the RNA sequence data. And of course, you have to look at them carefully and decide whether you trust what you're seeing. So you'll see, for example, these are 2 introns here that match what's in the gene model. You can click on these, and this will give you more information. For example, this had 22,000 intron spanning reads, so it's a lot of evidence supporting this particular intron. Same thing for this intron. You'll notice that the darker color indicates that it has more reads aligned to it. This has 25,000 reads aligned to it. Blue indicates that it's in the forward strand, and red indicates that it's in the reverse strand. So these are reads that aligned in the opposite orientation. And then you have this long one here, and you click on it, and you'll see that this has 136 reads, right? So it's not a huge number, or not a big number, but it's not a trivial number either. And so the question is this actually real or not? And of course, that's something you would have to investigate further. But again, based on the RNA sequence evidence that we have, this is what the algorithm is predicting. In most cases, if we zoom out a little bit more, in most cases, you'll see that hopefully those long ones are not really the norm. So yeah, so for some reason, this area here has some of these long introns being predicted, but in other areas, you won't see that. And so here's an interesting gene, for example, I can zoom into. It has predicted to have 2 alternative splice variants of this gene. And so you can look and see one of them has, in the 5' untranslated region, has an intron. And so you'll notice that in one case, the untranslated region has an intron, in the other case, it does not. And so these are the 2 alternative splice variants.
Okay.
Okay.
I just wonder if anybody else lost audio for about a minute. I guess we should just proceed.
Yeah, I don't. If you lost audio, not to worry. We have. I did not. So Jeremy is saying he did not. Thank you, Jeremy. So one thing that, as a reminder, this webinar is being recorded and will be made available both on YouTube and through GoToWebinar. And so if you miss something, hopefully you'll be able to go back and look at the recording. Okay, so So this is as far as I wanted to cover in terms of the JBrowse genome browser. We are definitely going to be moving to using this genome browser throughout our site and replacing the current genome browser. So I would encourage people who are interested in browsing the data in the way I was showing you just now. You can start playing around with it and getting used to how it works. It is definitely much faster and zippier than the Genome browser we're currently using. So I think overall it will be have a positive impact on using or browsing genomes. Okay, so again, as I mentioned, please add your questions whenever you have ones. But I will go ahead, Suzanne, and switch to you. So you can. So I will stop sharing, or maybe change presenter here and make you the presenter. Let's see if we can do it. Okay, so now you should be able to present and talk about gene page features.
Yeah, that sounds great. Thanks. Yeah, so there's two gene page features that are out in release 42 that I want to go over. The first is a Clustal Omega alignment tool that's in the orthologs and paralogs section. And the second is a new link out to a string tool. So I'm going to go to ToxoDB and demonstrate them there. And just as Omar did, I'm going to place a gene ID in the gene ID box and click the little micro magnifying glass to go to this gene page. And so now I'm on the gene page of lipoprotein. And I want to make it to the orthologs table. So I'm going to use the show contents section and click on number 7 down here, orthology and synteny. And that takes me to the data section of the page that deals with orthology and synteny. This table here, the orthologs and paralogs within EuPathDB, has 3 members. So this gene lipoprotein has 3 orthologs. And then down below here, down below the table are the parameters for this tool. So the updates to this tool are that we could now align protein, coding sequence, and genomic sequence. Previously, we only offered alignments of protein, and the genomic sequence can include up to 2,500 base pairs upstream and downstream of that gene. So to use this tool, I'm going to go ahead and check all. So I'm going to use this column within the table to choose these 3 orthologs to align. And I will choose to align coding sequences. And then another new feature is that the output format is available in 5 different formats. So we can do mismatched highlights, which is what we offered before, FASTA, Philip, Stockholm, or Vienna. So I guess I'll choose Philip, and then use this button here to initiate the alignment. I ran the alignment previously, so here are the results. You get the alignment in Philip format, and then down at the bottom of is a dendrogram that you can take actually to another program like ITOL or whatever to visualize tree. You probably have tree viewing software that you like. So that is is our Clustal Omega alignment tool, and these great updates to be able to align other types of sequences and get different formatted outputs. The next thing I'd like to show you is the string tool, and I'm going to use the contents here to go to that. I'm just going to type string in this search box up here, and I see that the string tool is in the protein features and properties section. I click that in the contents section, and I'm taken to that part of the gene page, right? So it doesn't look like much here. There's only 4 or 5 lines, but it's a pretty powerful tool. So the string is a database of known and predicted protein-protein interactions. So, and the tool that we've integrated here will submit your sequence to string where you can, you know, run their analysis on it and look for other interacting partners for this gene. So you have the choice of sending genes in from a Toxoplasma or its ortholog in Neospora or Haemondia. So I'm going to go ahead and use send Toxoplasma gene there. I'll click submit, and the first step it does is looks for sequence similarity of your gene to genes in its database, and it found 3 that it was similar to. And if you want to find interacting partners with these, you choose these one at a time and click continue. I'm going to choose a second one here and click continue. And the results come up pretty fast. So at the top, we have a network that you can, investigate, and it's interactive. And then down at the bottom, we have several tabbed pages of information. So here's the legend, and of course you can, there's a paper associated with string. There's lots of information here to be had, and you can delve deeper if you need to, or if you are interested. So are there any questions associated with this? Okay, great. I will turn the presentation back over to Omar.
Great, thank you, Suzanne. I see somebody potentially raise their hand. No. Okay. Oh yeah, I see somebody has a raised hand. Upati, let me see. I will go ahead and unmute you and see if your audio is working. If not, then I will ask you to enter your question in the question, or your hand went down. So I'll assume that you don't have a question. So,
I think I probably did.
Okay, sounds good. Not a problem. So again, as a reminder, raise your hand. We are monitoring that whether people raise their hands or add your question. I just did answer a question, for example, about the Genome browser, and so and whether you can save the visual representation. And so if you again, it's a good way to ask questions, and we'll go ahead and answer them. Okay, so the next section of this GoToWebinar today is we're going to spend time just quickly highlighting various data sets in our different genomic resources. So I'm going to start by highlighting data sets in FungiDB, and I'll switch back to Suzanne, and she'll highlight data sets in a few of our other resources. And then back to me, and I'll end by highlighting a data set in HostDB, which is one of our newer resources. So again, I'm looking, if I go back to the homepage of FungiDB, just to orient you, this just looks like ToxoDB or EuPathDB. All of our sites are designed under the same infrastructure and the same schematic, basically. So it should be easy to jump from one resource to another. And as you can see here, I'm just going to go to the news section. I'm going to select the latest news section for release 42. And so we're looking here at release 42. And as you scroll down, you'll notice that for fungi, we loaded a number of genome sequences and annotation available here. Actually, not all of these are annotated, and I'll describe this in a second, but most of these, so the Quaniella, the Naganitia, and the Quaniella, Fusarium, and Phytophthora are all annotated genomes. And then the Naganitia is actually just a genomic sequence without annotation, but it's still available for you to run various searches. If you ever want to go in and actually ask questions, well, I want to find all the genes from Quaniella because I want to ask some questions about them. What you can do is either you can go back to the homepage and look for the search that allows you to identify genes based on taxonomy or the organism, or you can mouse over this new search for genes. And I'm just using these tiered menus. I'm going to taxonomy, and I'm selecting organism. So this takes me to a search page where all organisms from that are found in FungiDB, in this case 129, these are all the annotated organisms, are available to you for here. So to select an organism, you can either open up these little arrows, which is pretty tedious, and you kind of have to go out looking for, well, where does my organism belong? Is it a citrid, or is it a zygomycete or whatever? So that has been difficult. But if you know exactly what organism it is, you can start typing, and as you start typing, it will filter. It's a W, I think. Yeah, there we go. it'll start filtering the organism names, and so you'll quickly see them. So here are the Coiniella genomes. If I select one of them or both of them, any of these, it'll select the organisms of interest, and then I can click on Get Answer. And so this is going to return all genes from the 2 organisms that I selected. So the result here is in the form of a search strategy. The step in the strategy is my search for genes from Coiniella, and you'll see that there's 17 or almost 18,000 genes in both of these organisms combined. If I scroll down here, you'll see the filter table, and you'll notice it says 0 for most of the organisms, and that makes sense because I only selected Coiniella. And then as you scroll to the right, at some point you will get to your organism group, and you'll notice here are both the Coiniella species that I selected, and you'll see that each have around 9,000 genes. And at the bottom, and you can, if you looking at this, if you decide, oh, you really want to look at strain 10.118 instead of and not strain 10.117, you can click on this little on the number here. It'll filter your results. It'll show you a little filter icon on your step, and now your results are filtered just for this particular strain of Coiniella. And now you can explore these genes, or you can add additional steps to your strategy and ask more questions. So let's go back to the news. Okay, moving down, there are a number of genome sequences where the annotation has been updated. So for some of our genomes in fungi, we have, and Tritrip and Plasmodium, there are curators available who actually on a regular basis update those genomes. And so some of these genomes have been updated, and all the information about what has been updated is available here. And also if you click on any of these, it takes you to the dataset record where additional information is available for you to figure out what is new. We loaded a number of transcriptomics datasets for fungi, and so there are 6 or 7 of them right here, and they span various fungal organisms. So again, depending on your interests, you may want to select one or any one of these. I'm going to go ahead and demo a search on one of these, and then I will come back and talk to you about the other datasets that are available. So I'm going to select this one from Fusarium graminarium. And so this is a transcriptome of heat-stressed Fusarium graminarium. If I click on this, again, it's going to take me to the dataset record, which has additional information about this dataset, including a link to the publication, if a publication exists. It shows you who the primary investigator is for this particular dataset, when it was loaded. As you scroll down, there will be a description here that gives you some idea about what this dataset is about, some more information about who else are contacts for this dataset. Moving down, you will see here there's a list of searches, and this I think is probably one of the more, well, it's a useful portion of this dataset record because it quickly lets you get to the searches that allow you to ask questions about this dataset, and I will highlight one of these. And then scrolling down, you will see that there's an example result from this dataset. Okay, so this is for this particular gene, and it's showing what the results are between the 2 different temperature growth conditions. And scrolling down, if there are important link outs associated with this dataset, they will be available here. So for example, you can go, this dataset was available in Geo, and so you can click on this link, it'll take you to Geo. So I'm going to scroll back up here. I'm going to select one of these searches. So I'm going to select this fold change search. And so actually, for the sake of interest, let's go ahead and select this differential expression search. And so if I click on this, it's going to take me to a search page, which allows me now, you can use some statistics here in your analysis, but let's say I want to find genes that are upregulated under conditions of heat shock, and so I can select, I'm going to compare the 37 degree time point to the 25 degree time point. I want to select things that are upregulated when I compare 37 to 25. I can change this differential regulation, let's change it to 5-fold. I can also adjust this p-value. So again, depending on how stringent or relaxed you want to be with the search, you can play around with this p-value. For now, I'll just leave it at the default, but of course, this is something you are welcome to change. And so once you click on submit, it'll give me the results. So it showed me that there are 1,798 genes that met these criteria. Notice that I have now a step in a new strategy right here, And my old one that I ran with the Cornella genomes is still here. So that's my second search strategy. If I don't really care about this anymore, I can click on this X here, which will close it, or I can delete it. And the difference between them is that if you just close it, it's not really deleted. It stays stored in your cache, in your browser cache. And if you log in, it will actually store it in your profile. And so that's a nice reason why one should log in, because any search you run will be stored in your profile, and you can always go back to them and re-examine what you did a week or a month ago. I'm going to go ahead and close this because I don't really want to look at this for now. And so you'll see that there are 1,798 genes that met this criteria. As I scroll down, you'll see here that it gives you the adjusted p-value. I can add additional columns here. For example, I can click on Add Columns. I can go to the transcriptomic section, Fusarium, and I want to look for Fusarium in this case. So you'll see here that there are a huge number of transcriptomic data sets, which makes it obviously hard to find. But again, with this filter table on the top here, I can just start typing Fusarium, and now anything with the word Fusarium in the title will start showing up. So hopefully this gives us an easier or a quicker way to access the data. So let me scroll down here and see if we can find it. So here are the heat shock ones. So now I can select, for example, in this case, I want to select, well, let's select all of these, or so let's select the 25 and the 37. And then what you do, so once you select these, what you can do is you can scroll down then either to the top or the bottom and click on Update Columns. And so now what's going to happen, it's going to load additional columns here. And those are what I selected were the expression values for each of these time points for 25 and 37 degrees. So you can see here that there's a huge number in 37 compared to 25. And then you can also add a graphical representation. So similar to the graph that I showed you in the dataset record page, you can load these here as well and view them. Okay, so let me go back to the home page here. So looking at the news, in addition to transcriptomic datasets, we loaded a couple of resequencing datasets. So these are sequences from field isolates that have particular characteristics. Again, you can go and look at these, and you can start asking questions about these datasets. So for example, this is a resequence data from Canada, ORIS. And so you can go down now and say, well, I want to find genes, for example, that have Snips in them. So here's, for example, identify genes by Snips, but there are many other types of SNP queries that you can run. So again, if this is something you're interested in, we'll probably have a webinar in the future which will be dedicated just to talking about SNPs and how you can search them. But for now, just a quick demo. I can go to identify genes by SNPs. This takes you to our generic SNP search page for all of FungiDB. You'll notice here the organism that's selected is Cryptococcus neoformans, so I have to click on this and select. In my case, I'm interested in Canada ORIS. And so now once you select this will filter the results, and it'll show you the data sets that are available for Canada ORIS. And you'll see here that there are 131 from this particular study. So I can either select these as is if I want to look for SNPs that are found in all the isolates, or I can filter them based on available information about the samples. So for example, I can say, well, I want to look only at the samples that came from India, right? So if I select this, it should filter out 16. So now I'm looking only at 16 of the 132 samples. And now I can scroll down, for example, and ask, let's pick genes that are in the Indian isolates that have at least, in this case, I'm picking non-synonymous mutations that have at least 10 non-synonymous mutations. And so once you click on Get Answer, this will give you the results, the genes that have at least 10 non-synonymous mutations from Indian isolates. And you'll see here that the answer was 0, right? And that's actually not a bad answer. It just means that based on the criteria I selected, none of them had this many non-synonymous SNPs in them. And so you can always click on the Edit button here, Revise, and then you can go in and modify your search and say, well, maybe 10 was too much. Let me pick 2, right? And you can keep playing around with this. The answer may still be 0, of course, and that's okay. That's something that you can go back and modify. So the answer was 8. So there are 8 genes that have 2 non-synonymous SNPs in them. When I compare the whatever 16 Indian isolates to the reference genome, and you'll see one of them is a multi-drug resistance protein, which would be, of course, of interest to look at. Okay, so I'm going to quickly go back because I know I don't want to take too much time here and switch it over to Suzanne, but I'm going to highlight one more data set in FungiDB, which could be of interest. So, and that is the phenotype data. So we are manually curating phenotypes and including them in the resource for some of the genomes. And you'll see here there's a manually curated Fusarium phenotype and there's manually curated Cryptococcus phenotypes. And so you can click on this and go to the information about this data set and then go and look for the phenotypes. But what I'm going to do, instead of starting the search from here, what I'm going to do is I'm going to go back to my strategies. And remember, I ran a search for Fusarium graminarium already, which is this one right here. So I'm going to click on this. And these are all the genes that are upregulated under heat stress, right? So now I want to ask if any of these genes also have an interesting phenotype associated with them based on the curated phenotype. So I'm going to add a step to my strategy. And based on this, I'm looking for genes based on phenotype. So you just have to find the word phenotype here. I'm going to click on phenotype evidence. You notice here we have phenotype evidence for many different organisms. In my case, I'm interested in Fusarium graminarium. And so I'm going to go ahead and select this curated phenotype option. And now I can pick from these different qualifiers, which are, again, as I mentioned, manually curated. So let's say I'm going to look for things that are, let's pick decreased amount, and let's pick virulence. Okay, so I'm looking for any gene when it was knocked out, it had a decrease in virulence phenotype. And I want to ask how many of these genes that have a decrease in virulence phenotype were also upregulated during heat shock. And so to do this, I will select this intersect option, right? So it's going to run one search here and then intersect the results of this search with my previous one here, which is in the background. And now when you click on run search, I got 106 genes that were manually curated as a decrease in virulence. 4 of them were found in the set of genes that were upregulated under heat shock. Now I can go down and look at them, and here's your list of genes. You can go visit them on gene pages or explore them further in many different ways. Okay, so I will go ahead and stop here. I think I covered everything for fungi, and turn it over to Suzanne, and she will cover some of our other resources. Okay, I see there are 2 EuPathDB organizers. Let me see if this is the right one. Okay, Suzanne, let me know if you didn't get, you got it. Okay, perfect.
Yep, here we are. Here we are at Plasmodia B, and we have a couple, some new data in here. I'm going to click over here in the news panel there to go to the news. And so the new data consists of one genome sequence and annotation. We added the Babesia divergens strain 1802A. This came from the Wellcome Sanger Institute, and this takes us up to 10 annotated genomes. So we're kind of proud of that. And then there are 2 functional data sets, an RNA sequence and a proteomics data set. The RNA sequence data set is the Theileria parva Schizont transcriptome, and that was put out by Joanna De Silva at the University of Maryland. This data is actually unpublished, but just as Omar was demonstrating with the RNA-seq search, that it's available to be searched. You can look at it in the genome browser, and so you can verify gene models and other ways to interrogate that data, right? So I'd like to go into the proteomics. We also have integrated a B. microti whole blood proteome that is from GI stage parasites, and that has recently been associated with a publication. So to go to that, I could click on this link here and go to the dataset page and get to the proteomics search from there, but instead I'm going to show you this where I can hover over new search and then go through this, navigate through these panels to get to the search. I'm going to look for a new search, search for genes based on proteomics and mass spec data. So then I'm taken to the search page, and the list of experiments that are available is listed here. I'm going to expand all, and we see that our whole blood proteome is the first proteomics data set that we have for Babesia microti. So I'm going to choose to search that, and our default parameters are very non-stringent, actually. So they're going to return a lot of genes, any gene really that's associated with its data set. And so when we received this data set, we received peptides that these researchers found in their analysis of whole blood. So we took those peptides and mapped them back to the genome, and anytime there was a match, then we associate that gene with this whole blood proteome. And this is what you're searching now, genes that mapped to their peptides. I'm going to look for genes that had, only one peptide sequence aligned to it. And when I click Get Answer down here, I'll find that 522 genes are associated with proteins in the whole blood proteome of Babesia microti. Now, this in itself is a good thing to do. You can, if you want to know what proteins are in the blood, but you can also, as Omar said, add this as a step to another search that you're doing. So if you want to find, if you have a set of genes like Omar did, 1,700 that were that he already had, and want to find which ones of those are expressed at the protein level, you can add this search to that strategy. So that is what we have added to Puroplasma. We also added quite a bit to Plasmodb this time. So here I am at Plasmodb, and I've already opened the News tab, and the new data in Plasmodb 42 includes genome sequence and annotation from a P. vivax-like genome. This is P. vivax-like is an organism that is discovered in African great apes, and it's thought to be the precursor of the human P. vivax, which we all know causes a lot of disease. So that's available. We have a new apicoplast proteome data set available. This is from Ellen Ye's lab, and this analysis used a combination of peptide proteomics, so laboratory proteomics, and bioinformatics tools to get a high confidence apicoplast proteome. I know this will be very useful to the community. So then we've also integrated some ChIP-seq data. This is from the Bartfi and Till Voss labs. What they looked at, it's a rather comprehensive study of the genome-wide distribution of heterochromin protein 1, HP1, which is involved in gene silencing. So they looked at different organisms, so Burgii, Chobody, Falciparum, and several others, and they also looked at different strains within Falciparum. And these are actually not searchable, right?
Sounds like we lost the audio for a second here, Suzanne. Don't know if you can hear us. So we'll wait for Suzanne to come back on. I think.
There is.
Okay, you're back, Suzanne. We lost you. We lost your audio for a few seconds. So you showed us the browser, but then we lost you. So maybe you can repeat what you said about these tracks. You said they were not searchable.
Right. So the data, there's no search for the data, but it's very visual. So if you want to investigate the data, you would go to the Genome browser, use the Select Tracks page to turn the track on, and then visualize these across the entire genome, right? So, and any place that you see a peak then would be an indication that HP1 is bound there. Okay. Yeah, my audio is fading in and out. It's a little.
That's fine. Let's keep carrying on. I think we now.
Yep.
Now your audio is good. So.
Okay, sounds good. So we also integrated two data sets of whole genome sequencing of isolates, mainly from PVivax and PVivax-like. This data is available, as Omar said, with for SNP analysis, and it's also available in our copy number variation analysis. And then we have some really interesting data, which I'd like to highlight, a protein microarray. So, and we can go to this record page. The antibody array of recent exposure to Plasmodium infection. And for this study, they took the serum of 186 individuals and scanned it on a peptide array that contains 856 peptide antigens. And it's like a microarray, so anything that lights up on the array suggests that that person made an antibody to the protein. So I'd like to demonstrate the search. If I go to New Search based on genes, down here to Immunology and Protein Array, I'm taken to the search page for protein arrays. And this one here, Antibody Array, is the new one. And I click here to go to open the search page, and I scroll down, and it's going to let me it's going to use the metadata associated with each of the patients whose serum was analyzed to stratify the samples before you run the search. So, for example, I can compare. Oops, I clicked on the wrong one. I'm sorry. Okay, now we're at the right search. Yeah. So here now we're looking at the metadata associated with this data set, the patients in this data set. And I can compare, I can set up two different groups and compare them, and then find genes that that differentiate between those two groups. So if I go to city, I can say I want to compare the 107 people here in Kahihi to the people in this township, looking for increased immunogenicity at a 0.05 p-value. I click Get Answer, and I see that I find 331 genes that are associated with increased immunogenicity in the second township. The data is represented in graphs and then different tables on the gene page, but it can be very powerful. Okay, so I'm going to go back to the news and see if we forgot anything. Okay, so if there's no questions about Plasmodb, we can move on to TritripDB, right? Okay, so here I am at TritripDB in the news section. And we have quite a bit of data this time. We've added 3 new genomes, the Bodo sultans, and this is great for comparison, sort of as a, you know, in your comparative genomics analyses. And the authors wanted us to make sure that you know it's a draft a draft genome and annotations, so take that in consideration when you're making, before you draw conclusions about things. We also added a Leishmania donovani CLSL strain, right? This is a PacBio sequence, and it's an isolate that causes cutaneous leishmaniasis. It was isolated in Sri Lanka. We've also added the Trypanosoma cruzi TCC genome. This is also a PacBio genome. Then we did, we added a new T. cruzi DM28C genome, and that brings us up to 3 versions of that genome. So we had to do a little bit of name swapping, right? So the newest one that we've added is a PacBio sequence, and was recently, there's a paper out about it just recently. So we've named that Trypanosoma cruzi DM28C 2018. This is our newest version. We had 2 previous versions in TritripDB, and we've renamed those according to the date that they were uploaded to TritripDB and appeared in the NCBI. So there's DM28C 2014 and DM28C 2017. 2017 is also a PacBio sequence, which is great. So we're also integrated annotation for Leishmania aminozensis. I'm sorry, I'm tongue-tied. But this is something that we worked on here at EuPathDB to help with the annotation, and that has recently been published. We worked closely with the authors to improve this annotation or update it. And so that's now available. That was a request. It's been a request for a while from the community, so we're glad to get that out. We have 3 RNA sequencing data sets, one that compares adipose tissue to bloodstream forms, one that's associated with amazonensis that we can actually integrate now that we have annotation for that, and another T. brucei RNA-seq analysis of different life cycle stages and bloodstream forms. So I'd like to demonstrate the adipose and blood tissue, and again for that I will use
Got really quiet again. I think maybe we lost Suzanne again.
some of them are associated with different organisms.
You're back again. We lost you, Suzanne. Sorry.
Okay.
So you got to the search page for RNA sequence. That's all good, I think. And so
okay.
Carry on.
Okay. Yeah. So as you can see, there's a lot of RNA sequencing experiments that we have integrated into TritripDB. The organism column here lets you know what organism the data.
So I'll come in and say, I think what Suzanne is describing here is that you can, in the organism column, you have the names of the organisms. So you can quickly, as she's showing, she's typing ADI for adipose, and it'll quickly filter the results and show you the data set you may be interested in. And I think that's really nice because as we get more and more data sets, it's much harder to find data sets. Okay, go ahead, Suzanne. I actually described what you were doing as you were describing it because somehow your internet is cutting out a lot. So.
Yeah, please just go ahead.
That's fine. I think I'm able to follow what you're doing and provide the voice.
Right. Yeah. So that little search mechanism up there is great, and you'll need to use that as we add more and more data. But here we are at the search page for this adipose tissue, and what they've done is they've compared mouse blood.
All right. So they compared mouse blood to adipose tissues that obviously contained parasites. And so what you can do is you can now compare samples, and you can decide what you want to find. So in this case, Suzanne is picking things that are upregulated. She's changing the fold change to 6-fold, and it looks like she's going to compare the fat samples to blood samples.
So I go down here and click Get.
Click on Get Answer, yes. Go ahead. So now
Yep,
you're back.
and click on Get. Okay, I'm sorry. So, and what we find is that there are 540 genes that were upregulated in blood versus, no, upregulated in fat versus blood at a fold difference of 6, and we're returning protein coding genes. So for each of these, then you can, it's always, you can download that list, but you can go on to really interrogate each one of these genes, and we can see that I do believe the data that I've been given, the search results, because here we have expression. This is a graph of FPKM versus sample for that experiment, and there's expression in fat, but none in blood, and that is pretty much the same all the way down. So let's go back to the news. It's a relatively recent discovery, or recently more highly studied, about adipose tissue, parasites in adipose tissue. We also have whole genome sequencing of isolates, a couple of T-Cruzi TLC1 isolates and clones. There is ChIP-seq data associated with KKT ChIP-seq. This is also important too, that we work closely with GeneDB and the annotators there. They actively curate the many of the genomes, and recently during the release 42 build cycle, they updated 495 genes in TB927 and 67 genes in Cruzi CL-Brenner. And these updates include many different things: product names, Go terms, EC numbers. They've added publications to those lists. So if you click on this here, you can go to a gene page that, or a document that summarizes that, if you're interested in what's going on with those genomes. We also get in user comments. Your user comments are very valuable information that feed our functional annotation. And during release 42, we integrated 48 comments from T-Brucei, and although only one from T-Cruzi CL-Brenner. And the last bit of data that we've added to TritripDB in 42 is from the TripTag project. And what we've done is added additional images and localization information, Go terms, that the TripTag project put out this month. And this takes our number of images to 5,292, and Go terms up to 4,954 for that project. Do we want to stop here, or should we look at the cellular localization imaging? We have 4 minutes left.
I think maybe.
Let's ask for questions.
Yeah, we should stop here, and I'll quickly highlight HostDB. And
oh yeah, great. Yeah, forgot about that.
Yeah, and then there's an answer a question. I might show something as well.
Okay. Oh, sorry.
I got it. Okay, so I think everybody should see my screen. So there was a question from an attendee about identifying 5' untranslated regions in a gene, and so this really depends. There are maybe 2 ways that you can get at those. One, is if it's part of the annotation, then we indicate those in a gray color. And so you can quickly go to a gene, and if you don't see any gray around it, then that's a good indication that potentially we don't have UTRs, at least in our version of the genome that we've loaded. So the other way of doing that is I'm going to use JBrowse to demo this, but I can go to the JBrowse genome browser. So this is, I was just going from a Plasmodium falciparum gene page, right? And see here are the genes. There's no gray upstream or downstream, so none of the UTRs are in the annotation. But I can quickly click on select tracks, and now I can, let's just pick, I'm just going to pick any, just one of these just to highlight this. And now let's go back to the browser. And now you'll see here that I have the RNA sequence data loaded. And what you can do is you can zoom in and see. So depending on, you may have to load more than one track of RNA sequence. It all depends on obviously the experiment and the time point that you're looking at. But basically what you do is you find the gene of interest and then see where the RNA sequence is dropping off basically and ending. And so the translation start site is would be right here at the start of this gene. And then anything, if we highlight, we can highlight this region. So depending obviously on the experiment, you could imagine that, oh, maybe this whole thing right here, anywhere there's any kind of transcript, is the UTR, right? And so that's one way of doing it as well. It's not ideal, but you're basing it on real evidence, basically, real data. And so, but I would recommend loading more than one experiment and different stages as well of expression. Okay, so I'm going to jump back to HostDB, and the two things I wanted to highlight here. One, the functionality will be the same, so really highlighting these is we don't have to repeat everything again, but I do want to highlight that we are loading more and more host genomes in HostDB and functional data from the host in response to parasite infection. So for example, in this release, we loaded the cow genome, the Bastard's genome, which was used in an experiment for infection with Cryptosporidium parvum. And so now, if you run a search using this data, so for example, I can take this full change search, and again, as highlighted before by Suzanne and myself, I'm going to pick, in this case, things that are upregulated during intracellular infection compared to uninfected or the first 2 hours after infection. And let's just pick the average here. So I want the average expression value for these 2 time points to be twofold lower than the average of these 2 time points right here. And I'm going to click on Get Answer. And so this returned 125 genes that met this criteria. You can look at them just as Suzanne said, so you can see the graphical representation of this. A nice thing you can do with any data in your result set is click on Analyze Results and run a Go enrichment. This can be done both on data sets that you've run in based on a question you asked in EuPathDB sites, or you can upload your own list of genes and ask for Go enrichment. And now you'll see here that the most highly enriched one, it's not a great enrichment, but it's enriched a little bit, and that is immune response genes, which is interesting in the context of infection. There are 5 genes that are involved in immune response that were upregulated in your set. You can click on this number 5 here. It'll run a new search and return those 5 genes, and then you can look at them and see what they are. It turns out they're all immune regulator genes. So this kind of makes sense in the context of the query we ran. Okay, so I think we will end here. We'll give you maybe a few seconds in case there are any last-minute questions. And otherwise, we will be running these webinars once a month or once every 2 months with our releases, so keep an eye for them. If there are specific topics that you really would like to hear, please drop us an email. You can always email us with questions by clicking on the Contact Us link from any of our sites. We're fairly good at responding. We try and respond within 48 hours, and depending on the number of questions we get. But please drop us questions, and any question is a good question, actually. We're happy to get them. All right, with that, thank you, everybody. Again, the webinar will be available as a YouTube or a video, which you will get a link to, and will also be available to others who could not attend this webinar. Thank you very much, and talk to you soon again. Bye-bye.
Bye-bye.